swap them. Okay, should be okay.
It's all good now. Thank you.
Good. Okay, thanks, Amy, a lot for organizing this course. I really enjoyed the previous talks, and I will, I think, expand a little bit more on some of the things that have been said, or give it maybe a bit of a different perspective, very much focused on rare diseases. And you will see that there's a lot of collaboration and synergies between the group that I lead and the project that I participate in and the EGA. So I'm going to talk basically or mostly about the RD-Connect GPAP, the Genome-phenome analysis platform. And we use this platform mostly, or the users use this platform mostly for diagnosing difficult rare disease cases, and also for gene discovery, always within the scenario of rare disease research, not so much in a clinical setting. So for those of you that do not know, rare diseases are rare in the sense that each of the rare diseases is affecting only less than 1 in every 2,000 persons, but collectively are quite common, as there are more than 7,000 diseases, and we estimate that between 3 and 7% of the population might be affected by one of these diseases. They are often chronic and life-threatening, and about 80% of them are estimated to be of genetic origin. So you see here the connection, the quick connection with the EGA and also with the GPAP, in which we are talking about genetics and genomes. So because each of these rare diseases are quite rare, there's usually a lack of diagnosis and a lack of therapies. And in fact, these are the two main objectives for the International Rare Disease Research Consortium. So improve diagnosis and also make more therapies available to the patients. And one of the issues is that because they are rare, we usually have a scarcity of cases that we can use to confirm diagnosis. So expanding our sample size is very useful. We also have a scarcity of data and samples to do research on. It's difficult to find this information and have access to it. And also, even if you know, we are able to develop a new drug or to repurpose a drug, we usually have a scarcity of cohorts for trials. Again, it's an issue of N, an issue of sample size. In that sense, sharing data and sharing biomaterials is very essential in this scenario. I'm going to focus mostly on the diagnosis issue, what in rare disease is called the diagnosis odyssey, or diagnostic odyssey. And this is that on average, a rare disease patient takes about 5 years to be diagnosed. Yes, some of them get diagnosed very quickly, but others are never diagnosed. On average, it is estimated that it is around 5 years. It is important to have a diagnosis, even if in many cases the treatment options are very limited. But still, by having this diagnosis, we can sometimes prevent progression, and we can predict how the disease is going to advance. We can also provide genetic counseling, prenatal or pre-implantational even. We usually reduce patient and family anxiety, because at least now they know what it happens, what disease they have specifically, and they can even join disease associations or patient associations, and also reduces healthcare costs, because you finally reached an endpoint, or at least a milestone in your ordeal, which is knowing the molecular diagnosis. But it is obviously challenging to find this molecular diagnosis, because as most of you know, a human genome has over 3,000 million positions, nucleotides. Actually, it's double because we have a diploid genome. And each of us has above 3 or 4 million variants. So identifying these 1 or 2 variants or mutations that cause the disease in a person is the challenge that we are facing. That's why a few years ago, we created the RD-Connect Genome-phenome analysis platform as part of an European project called RD-Connect. And what it is an online system that facilitates collation, sharing, analysis, and interpretation of integrated genome-phenome datasets for rare disease diagnosis and gene discovery. How it works is what you see on the scheme here on the left side. Basically, a data submitter, which is typically a clinical researcher or a clinician, submits on one side the clinical phenotype using HPO terms and other standards. And for this, they use the PhenoStore system developed in-house. And then on the other side, at the bottom, they submit the genomic data. This can be in the form of FASTQ, BAMs, or CRAMs. So we usually take the reads, the original data, the raw data, and we pass it through a data analysis pipeline. And then we integrate the results in the system, the phenotypic information together with the genetic information, providing means for the clinicians, the researchers to analyze this data. Also, all this data that we collect in the system, optionally, it can be submitted or brokered to the European Genome-phenome Archive, and I'll show you an example of that afterwards. The CNAG-GPAP is an ERDEC recognized resource. I mentioned before, ERDEC is the International Rare Disease Research Consortium. Currently, we have over 27,000 genome-phenome data sets, and these are mostly genomes and exomes, together with the clinical and phenotypic information. Not everyone, not all of these data sets belong to patients. In many cases, also they belong to relatives of these patients because it's easier to analyze the data when we have more information than just the patient. The data in the system is pseudonymized. It becomes available to all users after a 6-month embargo. So any user in the system at some point will have access to the data submitted by the other users. And this is what we are trying to promote, this data sharing, so that we can increase these sample sizes. The user registration is regulated by a data access committee, and we have a code of conduct and an adherence agreement that everyone needs to sign before joining the platform. The activity of the users is logged, and also we perform security audits periodically from an external company. You've seen in the previous presentations also that standards are key in our field of work. In our case, we use a lot of standards. Some of them are GA4GH, others are not GA4GH standards. And you can see here some of those on the side of phenotypic data. We use, for example, phenopackets, which has been explained before. But we also use the HPO for clinical symptoms, the Orphanet Rare Disease Ontology for clinical diagnosis, and OMIM for molecular diagnosis. On the side of genomic data, we use the typical FASTQ, BAM, CRAM, BCF. Most of them also now are supported or maintained by GA4GH. We also use Beacon, HTS-GET as a user, as a client, Matchmaker Exchange, and other APIs. We use AGNC for the gene nomenclature, HGBS for the bionomenclature, and our main annotation source is ensembled through BEP, the Barium Effect Predictor. So how does the system look like, the graphical user interface? Here we have a few screenshots of the PhenoStore, which is the system that we use to collect the phenotypic and clinical information from the patients with the rare disease and the relatives. And with this tool, we are using, as I mentioned before, standards, HPO, OMI, Mordo, and what we facilitate is a template or templates to facilitate this data entry, including a Pedigree editor. But you can enter the data individually using the click, the interface, but also you can upload the data in bulk in different systems, like using, for example, the PhenoPackage standard or JSONs, and even Excel files. And then we have also an API to be able to access the data in the system. The other key part of the system is the user interface of the genomics module, which is integrated also with the phenotypic data. And I'll go over these afterwards in the demo, so I don't need to spend time here. I'll just mention that we are about to release a new graphical user interface, hopefully by the end of the year, which is running in some of our internal platforms, but not yet in the RT-Connect system because it's quite complex, that one. And this will also come in with additional functionalities. In summary, there are quite a lot of functionalities in the system. So we have the standard filters, annotations for SMBs, indels, and CMBs. These evaluate the impact of the variants in the proteins, the frequencies in control populations within the database, predict in silico predictors, position of the gene, and genomic position, and so on. So these are basically annotations that allow us to establish some functionality on the genes. We have a lot of ways of creating genes of interest or in silico gene lists on the fly, real time. We have some which are predefined by our users, but others that come in through connections on the Genomics England Panel app, or Mendelian, or MIM disease, HPO symptoms, or Reactome. Basically, you enter a term, you enter a disease or a pathway, and then you'll get back all the genes which are related to this entity, facilitating then the filtration of the data. The system also has Exomizer integrated for a phenotype-driven strategy. The variants can be filtered by the ClinBio database, which is the one that collects the information about the pathogenicity or observed pathogenicity of the variant. Is a database in which the community uploads their results, their findings. We also have systems to filter by runs of homozygosity, and which is very useful in the cases in which we have consanguinity, because then we are usually looking at the smaller region of the genome, which is homozygous, and we can then make it easier to identify the homozygous variants in those regions. We have direct links to multiple external sources, and as part of the BASE project, we integrated DGenet, and we are still working on Foldex integration for the folding of proteins and how the different variants affect this folding, and hopefully this will be released with a new graphical user interface. In the system, you can also tag and share candidate variants using ACMG guidelines, so you can collaborate between different users to then indicate which variants might be of interest for further evaluation, or for collaboration or querying through, for example, Matchmaker Exchange, which is a system to connect different systems like this one. Also very important in a system like that, and I will not show this afterwards, the system allows you to search for variants in gene across all samples, because everyone is registered, authenticated, and authorized. You can search for variants across all the data sets in the system, something which is a bit different than Beacon, in which you are basically anonymously looking for some specific variant. And then we provide a collaborative environment. We're sharing data and queries, and also anonymize data discovery, as I mentioned, through Beacon and Matchmaker Exchange. Currently, we have about, I mentioned before already, 37,000 genome-phenome data sets. We have data from over 100 research groups and hospitals, and over 600 registered users. Registration is free for academics, although it has to go through that access committee to confirm that person can be registered. If you want to test it, and I will do afterwards, you can use the playground instance that we have in the RD Connect website. So I mostly talked about the RD Connect GPAP, which is the initial implementation of the system. But since then, we have also implemented and adapted the software, the GPAP, for other projects. So besides the RD Connect, which has data from many different projects, we also have an instance running for Navarra Biomed, in which we have been doing quite a lot of different projects, and also for the URDCAT project here in Catalonia, in which we have data from different hospitals in the region, usually of hard to diagnose rare disease cases, but also some undiagnosed cases from the CIBERON network, the CIBERON Enfermedades Raras. So I'll just briefly describe a few of these projects that we are working on. I'll try not to extend myself too much, so we have time for the demo. The first one is SolveRD. I think it was Babita, I think it was Aina who mentioned this one also. So this is one of the main rare disease projects in Europe currently, and it is heavily based on the ERNs. The ERNs are the European reference networks of expertise, clinical expertise on rare diseases. There are 24 of them, and you can see here that we are collaborating with 6 of them in this project very closely, and that they are centers distributed across Europe, contributing data and expertise into this project. Besides the European reference networks, we also have the undiagnosed disease programs from Spain and Italy working together in this project. What are we aiming to do in this project? We are aiming to solve rare diseases. Obviously, this is a very ambitious goal. And on one side, we are aiming to find causative genes in patients without diagnosis. And also, we aim to validate novel genes so that we have more information for the future to facilitate this diagnosis. In this project, we are collating existing data, over 19,000 data sets. We have, in fact, already gone beyond this milestone. and I think we are above 20,000 currently. And also we are providing 6,000 analysis slots to generate new data to hopefully crack some of these diseases. So we are providing whole genome sequencing, short reads and long reads, RNA sequencing, again, short reads and long reads, deep exome sequencing, which is useful for mosaics or for cancer samples, epigenomes, metabolomes, and proteomes. This is what we are trying to do in this project to facilitate the diagnosis of some of these cases. And then we have a few seeding grants that once we identify a good candidate gene, we can use them to functionally validate these results. We split the samples in mainly 4 cohorts. We have one cohort, which is what we call the unsolved cases, and this is where we are collecting most of the existing exome and genome data and basically trying to do reanalysis on this data to see if we can still get some diagnosis from existing data. And then we have cohorts 2, 3, and 4, which are different sets of specifications proposed usually by the ERNs. And what we are doing there is combining different omics data to then try to solve these diseases. So what is the infrastructure behind this project, which is what is most interesting here? It's simple, but yet complex. The way that we are working here is that we collect all the data from the users through the RDConnect GPAP using PhenoStore for the clinical and phenotypic information. We collect the raw data, we process this data through a standard pipeline, we generate the BCF and the GBCF files, and then we submit all this data to the EGA. So all the data, the raw data, the processed data, so the BCFs, together with the phenopackets, the PET files, and so on, goes to the EGA for further sharing in the future, internally in the project now, and also in the future once the project is over with the overall community through controlled access mechanisms. But also the data is available through the RDConnect GPAP. Then the data can be analyzed either through the GPAP, but also we can move data from the EGA to what we call the sandbox, which is basically an internal cloud infrastructure, and then we can do some analysis. And also we have some clusters which have been authorized by the consortium to download data from the EGA to perform more computer-intensive operations, because the sandbox has certain limitations and it's very expensive to have a very powerful system up and running. And then we have a big database that we call RD3, where this is data about data, which controls all the different data files and metadata and where all this information is at, including the results. Currently, from cohort 1, these unsolved cases in which we are collecting a lot of data, we have already 21,000 data sets collected. And from the analysis of the first 2 data freezes, which is about 7,000 experiments from 6,000 families, we have already solved about 1,000 new cases, which is about 15% solve rate only by reanalyzing data within the consortium and also by the groups themselves using the different tools that we provide and also their own tools. If you want to read more about the project, you can find a special in the European Journal of Human Genetics that was released June last year, with 6 papers explaining about the project and some of the cases that we managed to solve in this project. And also we are preparing a few more papers that will come out hopefully early next year. Another project I want to talk about in which we collaborate with the EGA is the European Joint Program on Rare Diseases. This is also a very large project. It's very different than Solverd in the sense that by itself it is not generating any data, but it provides grants for others to generate data. And you can see that the budget, total budget, is about 100 million euros. So it's very large. Lots of beneficiaries and also a big involvement from the European reference networks. In this project where the EGA and the RDConnect GPAP are mostly involved is in Pillar 2. And this is aiming to develop a virtual platform. What we are trying to do here is collect or connect different resources that are useful for the rare disease community, including registries, patient registries and biobanks, resources about cell lines and animal models, ontologies like the rare disease ontology or the human phenotype ontology. We are also bringing in toolboxes and help desk for clinical trials and translational research. We are trying to integrate access and privacy control mechanisms like Elixir AI, or now Life Science AI, and UPIT for privacy preserving record linkage, so trying to bring together data sets from the same individual, even if we don't know who that individual is. And then finally, as I mentioned, tools like the EGA, the Untitled GPAP, the Cypher, or cross-omics analysis systems. Another project in which we are heavily involved is the 1+ million genomes. And currently we are working there through the Beyond 1 Million Genomes project, which is a coordination and support action. In this sense, the project doesn't have any funds for development. It is more about planning and establishing proof of concept. And earlier this year, we established the rare disease proof of concept for rare diseases. And here we've been collaborating a lot between ourselves and also the EGA. Here, if you look at the bottom of the screens, you will see, I don't know if you can see my pointer or not, I'm looking at these two screen arrows that say start. There are different ways of accessing the system. And basically what we're trying to do is facilitate data discovery among the data sets in the system or in the whole network. And the way it works, this is through either a registered or a non-registered mechanism. So depending on the authentication and authorization level, you might be able to access or not certain features of the network and be able to access or not the data sets. And for this, and now you can go here at the top of the image where the blue boxes are, we have been working on several standards or implementing several standards, such as Beacon, the Elixir authorization and authentication infrastructure, matchmaker exchange, the GPAP, the federated EGA, together with APIs such as HTS-GET for data visualization. So you can see that all these comes into play in a very complex network in which have different access levels to the data. By the way, if you want to know more about this, I think there is a nice video in YouTube about this, and you can just search there, and you will surely find it. Another project, and I'm reaching the end of the presentation, that we are working together with the EGA is Genome-phenome for all. And this is a project in which we are trying to establish not federated data discovery, but rather federated learning. So artificial intelligence across distributed nodes. And here the main aim is to apply this to hematological diseases across different data sets in different hospitals in Europe. And you can see here, and this slide was someone, not exactly this one, but another one was shown before. The idea here is that we don't move the data to a central place, but rather move the queries to distribute the queries to different places and then collect back the results from these queries. So this is the rationale behind this federated learning. If you want to know more about the Articonnect GPAP, you can read the article that the paper that we published earlier this year. And also there are some training materials that you can access, and I'm gonna go through a few of those in a minute. On one side, we have the playground in the playground rdconnect.eu. And then also we have a synthetic data set that has been funded partially by BASE, which is at the EGA. And also again, if you don't need to write down the EGA study number, but if you look in the EGA archive search box for rare disease synthetic data, you will easily find that. And this is the data that we're going to be looking at today in the GPAP. So with this, I'll just thank and acknowledge everyone working in this project, obviously my team, but also many international collaborators, our users and testers, the European reference networks, and all the funders that have made this work possible. And I'll leave it here with the acknowledgement also to the BASE project. So maybe, Amy, we can stop for a second and take maybe a couple of questions if necessary, and otherwise I'll move to the demo.
Yeah, sure. Are there any questions from anybody in the audience? Feel free to write in the chat if you want. No? Otherwise, we can move to the demo. Thank you very much, Sergi. It was really nice. It was also very nice for me. Okay.
Hopefully it was not too difficult to follow. I know I spoke quite fast because I don't have that much time, but anyway, if you have questions, just put them in the chat. I'll try to answer them afterwards. Okay, let me see. Screen again. Okay, you can see my screen, right, again?
Yes. Yep, looks good.
Let's see how this works. Try to make full screen. There we go. Okay. Okay. So this is the main entry page for the RDConnect Genome-phenome platform. This is at platform.rdconnect.eu, but I'll make it full screen so we have more space for doing things. And here is where you can register if you want to register. Once you are registered, then you can submit data or manage your data, and you can access different applications for analyzing your data. What we're going to be doing in the demo is going to this orange button here and try out the GPAP modules. I'm going to go through one case, but you can do this afterwards at home if you want. This page, which has independent address, playground, rdconnect.eu, but as I said, you can get it from the main platform. Basically, has two modules for testing: the genomic analysis module and the phenotypic module. I'm just going to go into the genomics analysis module, and from there, I'll show you also briefly the other one. So the first thing that you need to do is to download the test cases, the examples that we have in the RDConnect GPAP, which are the same ones that we submitted then to the EGA. So you can go here and download these phenotypic descriptions. I'm gonna move into the screen now. And here you will see several slides with different cases: case 1, case 2, case 3, case 4. I'm not going to go through all of them, but you can see that you have quite a few cases to try things or use even in your training events if you want. It's good that if you want to do a training event, though, you let us know about it so that we make sure that the system is up and running and we don't plan any maintenance during that time. Okay, so with this, I'm going to start with the first case, case 1C. It is a male. It has 5 years. It was referred as a congenital metastatic syndrome. That is a neuromuscular disease with a congenital onset. It's progressing slowly, and the main clinical features is that it has neonatal hypotonia, distal arthrogryposis, inability to walk, and it has recurrent lower respiratory tract infections. And here on the right side, we have the drawing of the pedigree. We have the father, which is the ID is case 1F, and the mother with the case 1M, and then the son, who's a male and is affected in yellow, which is case 1C. You can see that none of the parents are affected of the disease, only the child is, and there is no consanguinity. So we could think about maybe a de novo mutation, something that the parents didn't have, but the kid does. We could also think about an homozygous case, like compound heterozygous probably. In the case of they are not related, but in some cases also could be that they are related and we don't know, and then we would have an homozygous recessive option. But we'll go straight away with the de novo option in the demo to keep things short. Okay. So we've seen the case, and now we have to log in into the system. To log in, we will use the username test and password 1234. But also you could use your Elixir login if you wanted to go through that, and then we would know who you are. We might enforce this in the future, but not yet. Okay, so we log in. And then we are presented with an interface in which we have basically 3 sections. The first one is where we select the samples and the filters that we want to apply with the data. The second is information about specific variants that we have identified or discovered in the family we are looking at. And then at the bottom, we have another table here in green that will tell us about the variants and the phenotypes of the individuals we are looking at. The first thing I'm going to be doing is select the samples that we're going to look at, case 1C. And then clicking in this plus button, I will create 2 additional samples. The mother is case 1M. These are the numbers I checked before in the slide, okay? And then case 1F from father. We said that we would try de novo mutation. So basically, we're going to be looking at mutations or variants which are reference alternative in the case, which is this one in the affected individual, but are reference in the mother and the father. That's why I'm selecting this. So basically, the case, the affected child has heterozygous position. The mother and the father would have homozygous position like the reference. This is very flexible, so you could build a family with many more members, several of them affected. You could play a lot with the different genotypes and have more than one check. But I'm just going through a very easy case. And also you could try compound add through this button. Then we have here some filters that I'm going to leave as they are by default. Basically, is the minimum depth of the sequencing to allow for a variant to be shown in the system, the minimum genotype quality of all these variants, and in the case of alternative positions, the minimum and maximum alternate allele frequencies, which tell us, gives us a bit of confidence that what we are calling it is indeed a heterozygous position. But if you're looking for mosaic positions and have low coverage, you might be able to tweak this a little bit because otherwise you might miss that. So I'm going to run this query already now, only with these filters. Amy, can you see well the screen? Is it too small? Can you maybe give me a bit of feedback there?
I see it okay.
Yes?
I think, yeah.
Excellent, good. All right, so you saw in that tab, we applied the cases and we applied no filters. And then at the bottom now, we got all the variants that meet this criteria. We have 18,000 variants. That's obviously too much data to go through if we want to discover a causative variant. So I'm gonna go and apply a few more filters. So I'm going to look at variants which have a high or moderate effect, which means that they are predicted to have some sort of effect to the protein. Okay? If they are lower modifier, in principle, they are synonymous changes or things that might not have an effect on the protein at all. And we'll start with being a bit restrictive and look for things that hopefully are or that might be changing the protein. Okay, and I'll run the query again. And now we already went down to 63 variants. So we have reduced a lot the number of positions because there are very few effects that might be deleterious. And remember that here we're looking at alternate positions, and maybe some of them, they are only doing an effect if they are in an homozygous state. More things that we can do, or additional things, is to look only for positions which are infrequent in the overall population. We have several filters for that. The nomad is usually the most useful one because it has more genomes and exons than any other filter. But also we have the internal frequency, which is a frequency computed from all the cases that we have in the system. But because we are at the playground, we only have, this frequency is not useful in this case. So I'm just gonna apply here, this 0.001 frequency, run the query again. And in this case, we only reduced a little bit the number of variants because already the ones which are expected to be deleterious, somehow they are usually also very low frequency in the population. And then here, when you select a variant, you have here at the middle window information, further information about these variants. So you can see the information from the different participants. What was the genotype? So as we were expecting 0, 1 and 0, 0 for the mother and the father, the quality of the genotype, the the depth of the sequencing specifically at this position, and the alternate allele frequency of this position in the case of heterozygous positions. But we can also go across the different tabs, and you will see functional information about these, the different predictors, the deleterious predictors that we have in the system, information about the frequencies of the variant. You can see that in this case, this is a very rare variant because it has not been found in the other data sets. Pathways in which this protein is involved, interactions with other proteins so that you can further expand the analysis. If there are cell lines, in this case, there is no cell line related to that one, so you might want to request this if there was one to make experiments, and so on. So you can see that there are different information that you can access from here. And then also from the bottom, there are lots of links that bring you to also useful information on Barsum or Franklin, or also, for example, into the GA4-GHP. Let me just look at that quickly. So this is Barsum, and this helps us to tell us, if a protein is deleterious or not, or pathogenic. In this case, the classification that we would get is uncertain significance, so likely, or it is difficult to know if it is causing the disease in this case or not. We could look at other systems such as Franklin, which has a similar approach. Again, here they classify this as a variant of unknown significance. So again, they are not able to tell us, or we are not able to confirm that this is a likely pathogenic or pathogenic variant, and then close the case. And we could also see the information in Beacon, and you could see that besides RDConnect, there are other projects that also are showing this variant, for example, in ICGC Cancer Project, but also Cosmic, also related to cancer and cell lines and so on. You have to think about Beacon. Also, an issue about Beacon that maybe it wasn't mentioned before is that sometimes you have the same data in several places, or you derive data from a data set into Cosmic or into other databases, so that maybe you see 5 hits, but actually it is the same individual or just a few individuals providing hits across different systems. But in any case, it's very rare, because you can see that I look for this position in many different sources. It only came up in a few of them, and some of them are just annotation databases. Okay, what can I do now to filter this even further? We can go to the again to the filters tab, and we can filter for, so we can bring in now the phenotypes into action. Okay, and what I'm going to do is use DGNet for that, and you will see DGNet a bit more later. But first thing I'm going to do is fetch the human phenotype ontologies, the HPOs from PhenoStore. I will show you these afterwards at the end of the demo. But I basically collected the phenotypic traits, the symptoms from this patient from the database integrated within the GPAP. And now, because we have these symptoms, we are going to make an extension or an association and see which genes would be related with this disease. And for this, I'm going to use DGNet. So I'm going to click on this DGNet button. And now DGNet gave me the genes which, according to the database, might be related to these phenotypes. I'm going to expand this a bit more. And I'm also going to go and search in Genomics Alien Panel Lab. I'm going to, instead of looking at phenotypes, now I'm going to look at the disease, right? We know it's a congenital myasthenic syndrome. So I'm going to look at congenital myasthenic syndrome here in Panel Lab. And I'm going to get also all the genes that, according to Panel Lab, which is basically a reviewed system, they are related to this syndrome. So now I have two lists of genes that I can use as a union to further filter my data. So I'm going to run the query again. And now I only get one result on, okay? And here, again, I could go and look at the position here, and here's a bit challenging to look at the thing and look at Beacon. Looking for different things. So we saw that the variant is in AGMD. This is a database that states pathogenicity of variants or gives us information about specific variants. And also we have it in UCSC, which, as you know, it's like a map viewer, a notation track also. So there's a lot of information there, and also we see it in RDConnect. So again, this specific variant is very, very rare. We only saw in 2 annotation sources plus in RDConnect. And then I could look into Barson and Franklin again and see what they tell me about this variant. Loading. Okay, in this case, Barson is telling me that this variant is pathogenic. It's quite likely with a lot of likeness. So it's probably something that we can believe. And also Franklin, in this case, it tells us it's likely pathogenic. So it is between likely pathogenic and pathogenic. And also it's telling us that it's likely that it might be causing disease. Furthermore, if we go to AMIM, for example, on the gene name, and we look in which genes this is involved, we can see that it's involved with several diseases like central core disease or myotonic dystrophy or King-Denver disease. So it is involved with diseases related to the ones that we are studying. So with this, I will finish the part on the genomic side, and I'll just briefly show you what you can see also on the phenotypic side. So I told you that we are integrating the phenotypic data together with the genetic data. So you always get a summary of the, so I have to make this a bit smaller. You get a data here, a summary in the main window about the family that you are looking at, the different IDs, the information. In this case, already RIR1 is selected as a causative gene, and also the different HPO terms and the diseases from these individuals. But also you could click on the PhenoStore ID, and it would bring you to the actual platform that we use to collect all this phenotypic information. I'm just going to go very quickly through this. And here you have all the information that we have in the database that we are then connecting through an API to from the other system. We have more detailed information about the case. We have the pedigree. You have the patient consent for matchmaker exchange or preventing communication and incidental findings, modes of inheritance, the medical history. You have plots on growth charts. If you enter the measurements in the systems, the signs and systems, the diagnosis, and so on. So there's a lot of information that you have there that you can get there. And also you could export to a Pheno packet, as I mentioned before, or to a JSON file, export it as a PDF, and so on. So it is quite useful to transfer data and to have communications with your collaborators. So I think I will leave it here. I just give a very shallow overview of what you can do with the system, and hopefully, if you are interested, you can go into the playground and explore a bit more yourself.